today via Zoom, and we can participate in this general lecture. So before we begin the main event, I would like to read the rundown for today's session. First, we will have the opening session, which includes a prayer and singing our national anthem, Indonesia Raya. And this will be followed by opening remarks by head of the Department of Biology, Prof. Sabto. And afterward, we will proceed to the main lecture delivered by our remarkable speaker, Ms. Novita Sari, Ph.D. from the A-Star Institute of Molecular and Cell Biology, Singapore. And lastly, we will have the closing session. So let us begin today's event with a prayer to ensure this event is guided with blessing. According to your own beliefs, let us pray together. Thank you. Next, let's sing together our national anthem, Indonesia Raya. And for this, please remain seated. Yeah. So next, to officially open today's event, we are honored to invite the head of Biology Department, Prof. Dr. Randa Sabto Purnomo Putro, MSI PhD, to deliver his opening remarks. For Prof. Sabto, time and screen is yours.
Yeah, thank you, Mbak Rasida, for nice being MC. Thank you very much. And of course, our guest lecture for today, Mbak Ita Sari, ya? Ita Novita Sari.
Yes, correct.
Okay, you're credited from our departments, right?
Yeah,
Yeah. At what time you graduated? What year?
2013.
Yeah. Ya, sebenarnya kita bahasa Indonesia juga bisa, but this is for making more comfortable and try to push our students to be familiar with international community, international events like this for today. I'm really glad that you are able to share ya, Mbak Ita Sari ya, for the most uh, cutting edge of the science in biology. In this case, is stem cell. You will mention about the new approach of the stem cell and cancer research, which is the really a good topic for today. we almost a week, within a week, we already introduced on some of the guest lecture, yeah, and then we always commit to improve our students to have a better understanding on the some interesting topics. For tonight, I think it's good to hear the stem cell and cancer research. It's something that has to be understood by our students, right? So thank you for Mbak Rasida. I will ask some the lecturer for today will come for the junior lecturer. Yeah, some of the student
Yes,
the
bro.
senior lecturer Pak Istadianto ya, Pak Sunarno. Yeah, there's some other senior lecturer. Maybe you still remember Mbak Ita,
Yeah.
those lecturer, and of course for the new junior lecturer also coming for today. Thank you for coming, Mbak Hernanda, Mbak Rizky, Mbak Rasida, yeah, and then some other, I don't know. I cannot mention one by one, but it's already 47 for today. So I think it's yeah, not many, but at least in one class, it's already there waiting for Mbak Ita to share her knowledge. As far as I know about the stem cell, yeah, it's a kind of a cancer stem cells, right? But they are in tumorigenesis. It's a kind of a subpopulation of tumor cells with the stem-like properties, yeah, and including self-renewal and differentiation. I think this topic today is very important to be having more understanding of new approach, yeah, in stem cell and cancer research, represent a new paradigm and to understand, treat, and manage the cancer, right? So by having more understanding about stem cell itself, we will offering the personalized therapy for example, more efficient in targeting drug-resistant cancer stem cell and minimizing side effect, yeah, by having more understanding. And then some other significance important for human health. I think the stem cell, understanding more about the stem cell, it's having more understanding to improve the cancer treatment, right? And of course, reduce side effect and other like preventing of the cancer recurrence. So in this case, this today and tonight, our students will having more understanding, and we hope that the students will be interested, yeah, in this topic, and then soon they will, look further to have research on that area. In Indonesia, I don't really know well about the office of the stem cell developed. As maybe I just mentioned one is Kalbe Pharma in Jakarta, maybe one of them, Mbak Ita, yeah.
Yeah.
Another one is in Bio Pharma. We have some alumni. One of them is Mbak Wirani Ayu. Maybe you know there. Maybe your junior or your senior, I'm not sure. But she's there already as a researcher there, and then she's been good in her career. It's almost PhD degree, and then some of the our alumni also working in some area. I'm not sure if they research on the stem cell, but living nowadays, it is a must to have more understanding of the stem cell, because some, our meals sometimes tend to be, you know, potentially induced the what we call cancer. We don't need that, but sometimes it cannot be avoided from the pollutants, especially that can induce the cancer in our body. So without further ado, I think it's better to hear our special guest for tonight. Yeah, thank you again, Mbak Ita, for sharing the information. Give it back to the moderator. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Prof. Sabto, for opening this event officially. So before we go to the main session or the lecture session, we would like to take a group photo. So please, all the attendants, kindly turn on your camera and please give your best pose. And I will take the screenshot for documentation in this evening events. Okay, please turn on your camera. Okay, I will take the photo or screenshot from the first slide. I will count in 3, 2, 1. And for the second slide, in 3, 2, 1. And for the last slide, in 3, 2, 1. Okay, thank you. Okay, without further ado, now we are delighted to move on to the main session, and I will hand over this session to our moderator, Ms. Rizky Sari, SSI MSI, to introduce our speaker and lead the session. So please welcome Ms. Rizky.
Okay, thank you, Ms. Rashida. Good evening, everyone. Welcome to tonight's session. My name is Rizky Sari, and I'm honored to serve as your moderator for this event. Tonight, we're diving into an exciting and important topic, which is new approach on stem cell and cancer research. As we know, cancer remains one of the biggest challenges in healthcare worldwide, and researchers are constantly searching for better treatment and hopefully cures them. Meanwhile, stem cell, often called the building blocks of life, or maybe we familiar as cell punca in Bahasa, offer incredible potential for repairing and regenerating damaged tissue. However, this advancement don't come without challenge. Question about how to responsibly use stem cells in cancer research and what progress has been made and what the future holds are key to understanding this field. So to help us explore this question, it's my great honor to introduce our esteemed speaker, Ms. Ita Novita Sari. Ms. Ita is currently a postdoctoral research fellow at the Institute of Molecular and Cell Biology, STAR in Singapore. She completed her Ph.D. in Bioscience at Sochunhyang University, South Korea, where she conducted groundbreaking research about polyamine metabolism in myeloid leukemia progression. With an extensive background in cancer biology, Ms. Ita has contributed significantly to the fields of leukemia, liver cancer, and cancer immunology. Immunotherapy, among others. Her research has not only led to high-impact publication, but also secured prestigious grants, such as the National Research Foundation of South Korea and Singapore's National Medical Research Council. In addition to outstanding research achievement, Ms. Ita has been recognized with multiple awards, such as the Young Scientist Award from the Korea Society of Toxicogenomics and the Best Postdoc Award from the Korean Society of Stem Cell Research. But what makes extra special tonight that Ms. Ita is one of our proud alumna of the biology department at Diponegoro University. So her journey for being a student here to becoming a leading scientist in cancer and stem cell research is not just impressive, but of course, it's inspiring of all of us. So today, she will share her expertise and inspiring journey with us. So please join with me in warmly welcoming Miss Ita Novita Sari. Miss Ita, good evening.
Yeah, good evening.
Okay, Miss Ita, thank you for joining us. So maybe before we begin our presentation, let's have a brief conversation first. Maybe as a proud alumna of Diponegoro University, especially in biology department, how does it feel to be back or to be reconnecting with the student and faculty here tonight, as virtually maybe?
Yeah, actually, it's a happy time for me. Like, I feel like having the nostalgic time because like it's almost like more than 10 years, I think. Yeah. But I sometimes I still connect with some of the lecturer in the Undip also and some friends also. So I'm really happy to be here today. So I hope that my experience can give you more like a little bit more knowledge to know about cancer biology and the stem cells biology. Yeah.
Okay, thank you,
Mbak
Miss
Ita,
Ita,
maybe
for
I
sharing that.
just one
Okay.
question dari situ Mbak Ita. Mbak Ita, you you've been having career in Singapore, right? For about maybe how many years you there?
In Singapore is now 2.5 years.
Two
Yeah.
and a half. Before that,
Before that, I did my master Ph.D. program in Korea and then 1 year's postdoc, so total for 7 years there.
wow. Yeah, big applause for our alumni. It's really we happy with the alumni that has been successful, yeah, for the career. But just hope, really hope that you attract our students and graduates to come over there, be selected. Maybe just propose for the opportunity for the alumni to have, a chance to work as you do. We expect that maybe if this is there is chance, please let me know, and then we will inform the graduates for the excellent graduates to follow you. Maybe one or two, maybe someday. I don't know if this is possible. It's really great. Thank you.
Yeah, actually, I'm really happy to share and for me myself also I really encourage younger generation also to have higher education also, and I'm not saying that education in Indonesia is not, I mean in the biology fields is not really supported actually, but yeah, I do recommend please keep trying to improve ourselves and then learn outside also, study abroad also. There is a lot of information from the scholarship, so anytime you have any question you can contact me, then I can share some information for sure.
Wow, that's very promising. As students, we must hear and ask the question as many as you want. Okay, back to Rizky. Thank you.
Okay, thank you, Prof. Sabto, and thank you, Miss Ita, for sharing that. It's really heartwarming to hear you feel about reconnecting with your alma mater and, of course, Diponegoro University. Maybe some of us already inspired with your journey, especially to the student who are just starting their journey. So maybe let's move on to our main topic. But before we begin, let me inform everyone about our schedule. Maybe the presentation is estimated to take around 90 minutes and followed by question and answer session. So if you have question during the presentation, feel free to take note of them and you can share them during the question and answer session. So let's make tonight an interactive discussion. Okay, let's move on to the main topic of tonight's discussion, your approach on stem cell and cancer research. So, Miss Ita, the time is yours.
Thank you. I will share my screen at first. Okay, can you see my screen?
Yes, it's already seen.
Okay. So again, thank you so much for inviting me. And It's an honor for me to talk to you today and to share my knowledge about the new approach on the stem cells and cancer research. So now I'm currently working in the Singapore at the Institute of Molecular and Cell Biology of A-STAR. And this is the outline of my presentation. So first of all, I would like to give a little bit introduction of the cancer and then cancer stem cells. So, and then the second, I will give you a bit like background about my previous research experience during my PhD study, as well as my ongoing research project in the A-STAR, which is involving the advanced study for the cancer stem cells using the single cells and then spatial transcriptomic analysis. So let me start with the first part. So I just give you, I try to make it simple, but I hope you can understand the principle of the cancer and cancer stem cells. So there's a multi-step tumorigenesis that is happen from the normal cells to become a cancer. So it will start by the initiated cells, which is one or two cells, which is they're having the outgrowing or hyperplasia of the size of the cancer of the cells, and it will also involve in the influencing of the surrounding cells or microenvironment to make it's more like hyperplasia or more bigger size and growth. And then during the time, it will become bigger and bigger, and then it's become a mass or a solid cancer tumor. And then it will have the aggressiveness and as it can invade to the other organ of the body, not only from the primary tumor, which is we can call it as a metastatic event or invasive of the cancers. So depending of the type of the cancers, for example, cervic cancer or breast cancer, colon, prostate, they have different time where they can develop from the normal cancer, normal cells to the cancer cells. Some of them can take like 6 to 10 years or 5 to 13 years, or some of them like leukemia, for example, it also can take only like 3 months or 6 months to develop into the aggressive cancers. So the growth of the cancer cell actually is affected by the multiple process or multiple factors, which is we call it as a hallmark of cancers. So for example, why we call it cancer cell? Because it's have the ability to overgrowth, or they doesn't have the capability to suppress their proliferation. That's why they are keep proliferating. So they are unable to avoid the immortality, as well as they have the capability also recently is to avoid the immune system. So once the immune system actually is function to destroy the strange cells from our body, but however, these cancer cells are having the ability to inhibit this immune cell recognition. So that's why they are become more grow and then more invasive to our body. So the growth of the cancer is also affected by some of the existence of the proto-oncogene, which is the gene that can cause the progression of the tumor, and also the disability of the tumor suppressor genes to inhibit the cancer progression. So what happened is like mostly of the tumor suppressor genes, they will have the loss of function or loss of the heterogeneity, so that they will lose their capability to inhibit the cancer. And then this mutation will cause the cancer cell to grow even more mass. And there is also proto-oncogene, which is the expression is also depending on the cancer cells, what type of the cancer cells. For example, in the breast cancer, like HER2 or BRCA1 mutation is the prominent one in the breast cancer. And then when it's overexpressed, it can cause the more aggressive cancer progression. So the balance of the oncogene and then tumor suppressor is very important in order to maintain the to inhibit the cancer progression. So as we know also, like the sometimes like these cancers not only grow in the original place where this primary tumor is grow, but they also can invade to the other place outside of the original primary site when they reside. So this primary tumor, they will have the ability to invade the blood vessel. So it will start with the intravasation in here. For example, this is a solid tumor. It has the possibility to invade the bloodstream and enter the blood circulation. And then it will go to the specific area. For example, if we have the colon cancer, then it will reach, for example, in the kidney or heart or the lung. And then it will create the new environment in the new site of the metastatic. It will have the it will form the small formation of the cancers, and then it will make a big mess also in the secondary tumor sites. So we call this event as the metastasis. And many factors is also involved in this metastasis. For example, like there's some epigenetic factor or some soluble signals such as like cytokine or growth factor, or also the presence of the some microbiota in our gut is also affecting this metastasis event. And also from the cancer cell itself, there is a change in the adhesive ECM component as well as the some of the pressure of the cells that can induce this metastasis and invasion. And in the cancer metastasis, there is a specific event that is very famous for the solid cancer propagation, as well as for the, mainly for the solid cancer propagation. So we call it as the epithelial mesenchymal transition. So in the primary tumor, usually this cancer will grow in the epithelial cells. However, when they have the ability to move to the other place, they will lose this capability, they will lose this characteristic of the epithelial cells, and then they will be more mesenchymal cells. And then this mesenchymal cell will grow and then circulating to our body. And then once they migrate together, they will come back later again as the epithelial cell. So usually we call it as the EMT progress when they are starting to migrate. And then once it's reach the secondary place of the growth, we call it as the MET progression. And yeah, so this is the process of the EMT. Same with the invasion. So it will extravasate the blood circulation, and then it will go to the secondary part of the body. Generally, specific type of cancer, they will have specific place where they can metastate. For example, in here, like kidney cancer, they will have the probability to metastasize to the lung, liver, and bone. And then the ovarian cancer will go to this specific area, as well as from the breast cancer, usually we can find can go over to the bone, lung, liver, brain, and etc. So they have specific place, like they are happy to grow, and then they choose the place, and then they will grow in the specific sites. So many effort has been invented to inhibit the metastasis process. So what happened is that during this metastasis, many of the signaling pathway is actually involved. So for example, like the MAP kinase pathway or the KRAS or BRCA mutation is also very common one in the cancer. So mostly people will try to inhibit this pathway in order to inhibit the invasion and metastasis of the cancer. So we call it as the targeted therapy because we target specific pathway of the cancer growth. However, as we talked previously also, there is a stem cells. We know that the stem cells actually they have the capability to do the self-renew, where they can self-renew themselves. They can make the expansion. For example, in here is the hematopoietic stem cell, but it also can be applied for any type of the stem cell, for example, embryonic stem cells. But it's also in the same time, not only in the self-renew, they can also have the capability to go for proliferation and also differentiation to the specific mature cells. And it also, some cells will also undergo further death or differentiation. However, in cancer also, like in the normal cell, the cancer stem cell, they also have the capability to self-renew themselves, and then they have the stemness property, which is make them more difficult to treat, and then they have more clonal expansion to multiply themselves, and then it's very hard to be targeted by the drug or the and they are mostly like resistant to the therapy. So this cancer stem cell is already exists since like early 20th century, and then it's still until today it's still become the major concern of the cancer treatment. And then this cancer stem cell usually they have specific markers. For example, in the leukemia, they have the specific this cell surface marker that we can recognize them as the cancer stem cell population. And this cancer stem cell usually reside together with the microenvironment of the cancer cells. And then this cancer stem cell usually they will evade from the drug inhibitor. So they will, once the normal tumor cell they will die. But however, this cancer stem cell usually reside in our body, and in the future it will grow again to the differentiated cell or to the stem cell progeny cell, and then it will create more prone cancer progression, and then make the new formation of the cancer cells. So yeah, so there is so many of the cancer stem cell marker that is like already well known also. For example, in the solid tumors, here we have the EpCAM, CD44, CD133 is common for the colorectal cancers, and then the hematopoietic malignance, for example, in here like the CD34, CD25 is common for the leukemia. And then this MUSA-C2 and BMI1 is also like stemness factor that is also famous to induce the cancer stem cells in both solid tumor and then the hematopoietic malignance. So why is it so difficult to target the cancer stem cell? Because these cancer stem cells, they have the capability to have the dormancy property, which is they can stay as the quiescent stage where there is no proliferation, because mainly of the anti-cancer drug, they will target the proliferating cells. However, these cancer stem cells are dormant because they have the stemness property. So this drug cannot target the cancer stem cell. That's why they keep reside in this body, in our body. As well, they have the anti-apoptosis capability, and then they can also induce several conditions like hypoxia and influencing the metabolism to keep the sustainable of the cancer stem cells to keep reside in the body, and then in the future they can grow into the secondary tumor. So similar with metastasis, also several efforts already made for the treatment of the cancer stem cells. For example, by targeting the stemness differentiation capability, as well as the metastasis capability of these cancer stem cells. And recently also, like there is many effort of the immunotherapy where we can use some of the cancer immune cell to target the population of the cancer stem cells. For example, in here, NK cell, or for example, T cell, DC cell, people are using the CAR or chimeric antigen receptors to start to intervene the cancer progression. So mostly they are working by targeting the checkpoint inhibitor. For example, in the T cell is PD1 and CTLA4, which is the this will bind to the PD-L1 receptor, PD-L1 ligand in the cancer cells, and where it bind to the cancer cells, this T cell will recognize it and then will cause the apoptosis of the cancer stem cells. So many efforts are still developed in this area for now. So this is a bit introduction of the cancer and cancer stem cells. And So I will share to you a little bit like the common methods that we use or we use to study in the cancer and the stem cell, for example. So mostly like we will divide the study into the 3 part or 4 parts. For like it will involve the in vitro, or we culture the cells in the 2D or 3D dimension, and then later we will go to the animal model using the mouse or rat. So we call it the in vivo model, and then we also use the ex vivo sometimes, where we take out the sample from the cancer patient and then we grow it into the mouse, as well as we use the PDX or patient-derived xenograft. Also, it's the same, and usually we will correlate this result in the human clinical samples. And then the last but not least, also we will try to use the inhibitor of the pathway to implement our inhibition in the in vitro and in vivo, as well as in the patient sample. So in the in vitro study, usually when we have interest of the gene or of the signaling pathway or the specific microenvironment, then we will create the genetic modification of the gene, for example, gene X. Then we will try to modify how if we reduce the function of this gene, or how if we give the more expression of this gene, what will happen to the cell. So what we do is we have the loss of function assay, where we genetically make the construct of the plasmid to have the knockdown of the this gene by ssRNA or short hairpin RNA, or siRNA, which is the short interfering RNA, which is its function to reduce the expression of the specific gene. Or we can do the CRISPR knockout also, which is also similarly we can use this to remove the expression of the specific gene. Or we can do the site-directed mutation, for example, for the gene that is important for the specific enzymatic activity or specific binding, we can make the mutation, and then we can try to put this construct to the cells and see what happened to the cell if we reduce the expression. And then we also do the gain of function, where we put more expression of this gene, and then we see the progression of the growth, for example. And then sometimes we can do also the knockdown and overexpression, which is we try to rescue after we remove this expression, and then we put back the overexpression construct of the gene. What will happen, whether will they back to their original characteristic or no? So the assay for the cancers usually, like, because cancer cells they have the overgrowth, they have the hyperproliferation. So first of all, we will check this knockdown or overexpression in the growth assay, for example, like the proliferation or colony formation, where we can see like the clump of the cancer which is getting lower or higher. And then if we want to do the metastasis analysis, we can do the angiogenesis assay, which is to see how this cancer have the capability to invade the blood vessel, as well as for migration and invasion, to see how the capability of them to migrate to the other area. And then this is some of the common pathway we use for the pathway analysis, as well as, we will try some of the drug inhibitor to this cell culture, and then to see how it's affect how the effect of this specific drug to reduce the progression of the cancer. And the second one, usually we will do the in vivo study, where we have to use mice, and then usually we inject the cells from the mammalian cell lines, or can also from the patient-derived samples, which is we already knocked down or overexpressed, and then we will inject them to the mouse, which is, there's several type of the injection, like subcutaneous means like you will inject the cancer cell to the surface of the skin, and orthotopic is the where you can find where you need to inject the specific cancer cells to the specific site of the body. For example, if you want to inject the breast cancer, then you inject on the mammary gland of the mouse. However, the subcutaneous can be put it everywhere, like maybe in the flank or in the back, something like that. And the other is the intravenous injection. We do the through the tail vein or through the eye, and then we can see like how this cancer will grow later in the specific area of the body. And for the metastasis also, so for example, we do the injection in the surface of the skin of the mice, and then we want to check the metastasis, like will it grow to the other part of the body. For example, if we inject the colorectal cancer to the skin or by IV injection or intravenous, so later we will open the area, for example, like the heart, lung, and then kidney, to see whether this specific cancer are able to grow in the specific secondary site. And also, yeah, currently also many people are also using the knockout mice, which is we delete specific gene from the mice, and then we see also how if we induce the cancer in this mice, will the cancer still grow or not. Also, yeah, as I mentioned previously, we're using the patient-derived sample and then inhibitor analysis. And lastly, we will correlate it with the human data, where we can use the patient-derived sample. For example, if I want to study about leukemia, I will get the blood cancer sample from the patient, and I will modify them, and then I will do this assay similar with the in vitro one, and then I will also can inject these samples to the mice. And finally, we will also check the correlation or survival analysis by bioinformatic analysis to see like whether our gene of interest actually is involved in the survival of the cancer cell or no. So this is very important, because if we found our gene of interest, actually when it's highly expressed, it can cause the more prone progression of the cancer cells. Then we can use it as a biomarker of the cancer, and then in the future we can propose it as the target for the development of the drug inhibition of the specific cancers. So this is overall for the first part. So I will give a little bit I will share a little bit about my previous research in the PhD study. So during my study, I am studying in the Sun Chun Yang Institute of the Medi Bioscience. So actually, I just a little bit like I just start for master degree at my university in here. However, like in the meantime, I feel like it's okay. This cancer field is very interesting. Cancer stem cell is very interesting. And then my professor actually advised me to change to the MS PhD program. So that's why I take 6 years until I graduate from the PhD. And so yeah, 6 years from MS to PhD program. And during this time period, so I spent the whole of more than half of my research was focused in the leukemia study. Meanwhile, some of them also I study about the colorectal cancer, and then also I study about several of the solid cancer, for example, the lung cancer, liver cancer, as well as the liver fibrosis also. And also I study about the immunotherapy. So this immunotherapy is like usually I use the NK cell or CAR T cell. So I will isolate the this immune cell from the peripheral blood from the normal patient, and then we will modify them and then try if we target specific gene in the solid tumor or maybe in the leukemia, can we use it like in the clinical in the future, something like that. Yeah. So yeah, it was I was very lucky because my PI was really care about me. That's why, yeah, we have several publications together so far. So let me share you a little bit about the hematopoiesis. So hematopoiesis mainly reside in the bone marrow. And then this hematopoietic stem cell actually is also reset together in the bone marrow together with a specific cell type. For example, in the red blood cell, white blood cell, platelet, and others. And this blood stem cell, it will grow later to the myeloid stem cell and then lymphoid stem cells, and then it will grow to the specific differentiated cells, which is it will have their specific roles in our body. For example, the red blood cell, it will carry oxygen or other substance to all the tissue of the body. Platelets will form the blood clots to stop the bleeding, and white blood cell to fight infection and the disease. So in this one, hematopoietic stem cell or SSC is also similar with other stem cells. They have the capability to do the self-renew as well as to have the multipotent capability to differentiate to the other type of the stem cells. So they have the symmetrical division, which is they will differentiate, they will self-renew or expand to the same cell type, or they have the ability to do the asymmetrical division, where they can grow into the other specific cell type or stem cell type. And so during this differentiation, so actually the hematopoietic stem cell, they need to go to the specific type of the blood cells where it should be where it should have their normal function to our body. So what happened during the cancer is that there should be a balance between the self-renew and differentiation of the SSC. However, once this balance is disturbed, it will cause to the leukemia progression. So leukemia, or as we know as the blood cancer, is a cancer from the blood. It's arise from the blood stem cells, and then it's mostly known as the white blood cells cancer. And during my study, actually, I'm focusing a lot in the chronic myelogenous leukemia, which is the mostly common type of the blood cancers. It's caused by the translocation of the ABL and BCR chromosomes from the 9 to the 12, which is it will cause this abnormality. And then this cancer, it will grow from the blast to the blast crisis, where if we have this type of the blast crisis, it only takes us like 3 to 6 months for this cancer to like kill someone. So many research are really invented in this blast crisis study. So why I studying this one? So previously my PI is one of the author for this Musashi-2 binding protein in the myelogenous leukemia. So actually he found out that this gene is very important for the CML progression as well as the AML, and high expression will cause the cancer cells to become more aggressive. And also one of the Musashi target is the tetraspanin 3. It's found to be also important for the AML progression. So that's why I'm really curious what is the other target of this Musashi gene, which is will have the capability to affect the CML progression. So yeah, this is I just share give a little bit background like how I found this one. So we did the RNA immunoprecipitation, and then we found that this gene, which is called the AMD1, have the high connection with this Musashi-2. It's bind as the Musashi-2 binding target. And shortly, like this AMD1 actually is a key enzyme in the polyamide biosynthesis, which is important in the RNA, DNA, protein synthesis, and it's really important in our body to maintain the normal transcriptional process to have the normal metabolic capability. But however, in cancer, actually, we found it to be high in the BCCML. So like what I said previously, what we can do is like we can modify the we can modify the gene, for example, like the knockdown, like so in here also we use the shRNA to knock down this AMD1 gene using the shRNA, and we check the mRNA expression, we confirm that it's knocked down, and then we check also on the capability of this gene to involve the colony forming ability of the normal stem cells, as well as in the BCCML or in the blast crisis leukemia. So once we confirm that the gene is having specific role, like once we knock down this gene, actually in the colony formation is reduced, means that this gene is actually important for the growth of the cancers. So furthermore, we go for more characterization of the function of this gene. I will not go to this detail. And then another example is also we do the overexpression, which is we put the more expression of this AMD1 gene, and then where we can see in here actually like when we put this gene in the high expression, we can see that compared to the control, it has more expression, and then confirmed by the Western blot also in here. And we do also the rescue experiment using the knockdown as well as putting back the overexpression factor to this cancer cells, and then we see how the colony forming ability, as we can see that once we reduce the expression or we knock down, we can see the less cancer formation. However, when we put back this gene expression to the high one, it can rescue the proliferation of the cancer cells. So it's confirming the importance of this gene. Yeah. And also, the next step is like we want to confirm how this gene actually important, not only in vitro but also in mouse. If we knock down this gene, will it also reduce the cancer formation of the blood cancer in the mouse? So we created the condition of this mutation from the BCCML, which is the BCR-able, and then additional this translocation for the blast crisis step. And then we also put the knockdown of the specific gene. For example, this one is AMD1. I injected the cells to the first mice, and then the secondary mice, we will inject this to the secondary mice. And after that, we can see like how the survival. For example, in here, the black one is for the control, and then this one is the dash one is for the SSAMD1. We can see that the progression of the primary transplanted mice, actually the cancer is reduced when we knock down this gene. And when we put in the secondary mice, also actually is to check the metastasis actually. And then we found that when we knock down this one in the secondary transplanted mice, it will reduce the progression of the cancer in these mice. So it's a good sign. Yeah. And we also try to do some rescue experiment by knocking down and putting back the expression. And then similarly, we see like then when we putting back this expression, we can see like the mouse is going to death also during the 40 days. Yeah, so and further to usually we also go like by specific approach, like if we found this gene is important for the cancer progression, then what signaling pathway is important, what signaling pathway they are affecting in order to promote the cancer cell progression. So usually we will do the RNA sequencing, and then we analyze what is the gene which is affected, for example, TNF in here, as well as respond to the inflammatory response and other. And yeah, they will involve several of the bioinformatic analysis. And finally, actually in this finding, we found that this gene actually important for the oxidative stress response, as well as the ER stress and UPR unfolded protein response pathway. And also, we know that some of the cancer, they can have the capability to be resistant for the drug treatment. So we also created the cell line, which is the resistant for the drug. For example, imatinib and nilotinib is for the leukemia. We check their expression also, and then we also try to do also the same knockdown experiment, and then we found in here also like this knockdown of the gene AMD1 can possibly reduce the colony formation of the BCCML cell line. Yeah, not only in that one, also we check in the several of the drug resistant cells, and we get the consistent results. So far, like it's confirming our result that this AMD1 gene actually important for the BCCML, and then more importantly, actually, it's important also for the drug resistance of the cancers. And also, more importantly, we correlate this result with the clinical data. So we get the patient samples from the hospital, and then we check the expression of this gene, and then we found that this AMD1 gene actually is having high expression in the last stage of the CML progression. And then when we conducted the knockdown of this AMD1 gene, actually, it also can reduce the colony formation of the patient sample. And when we check the survival of this patient in the real time, actually, when this AMD1 gene is highly expressed, then the patient is dying. The patient is dying in the more shorter time compared to when the expression of this gene is low. So we always try to connect it on the human patient because of what? Because the research which can be translated to the clinical is more important, because in the future we can create like some drug inhibitor or antibody inhibitor to inhibit this specific gene so that we can eradicate the cancer. So the same thing also, actually we also conducted the drug screening for this AMD1 gene, and then we found this specific compound AO476 is a specific inhibitor for AMD1, which can inhibit also the myeloid leukemia progression in the BCCML or the chronic myelogenous leukemia, and then the AML. So it shows that the growth of the cancer patient-derived cancer cell is reduced after we treat with this drug. Yeah, and then for the validation, we also check it to the mouse. We inject the cell to the mouse, and then we see in the mouse how it's grow. And then when we see this, so we inject the human cancer cell lines, and then we treat them with the drug. And then we see human CD45 marker, which is the marker for the human blood cells. It is reduced in the mice that we are treating with this specific inhibitor compared to the control one. And more importantly, also when we treated the drug should be safe to our bodies. It shouldn't have any cytotoxicity to the other sides of the body. For example, in here, in the lung and the liver, we also check like within the control and then the drug treatment, whether it have the cytotoxic effect or no. But luckily in here, like we found that this drug is safe to be administered. So yeah, so far we have patented this drug, and we are working with the company to generate this drug more, and then they will try for the clinical trial. So that's overall all for my previous research experience. So this is the last part of my talk. I will talk about the ongoing research project in A-STAR of Singapore, which is mainly I'm working for the single-cell RNA sequencing and spatial transcriptomic to study the stem cells and then the cancers. So for the stem cells, mostly I am working with the embryonic stem cells, which is this embryonic stem cells will happen during the embryogenesis, which is the starting at the E9.5, it will start to form the body of the mice, and then during this time they will grow to the adult mice also. And as well as during this embryonic development, actually the hematopoietic stem cell is also grow during this period. So it will start growing at the embryonic days, start from the 11.5, and then it will mainly support the hematopoiesis process of the fetal liver. And then once the mice is born, it will, this hematopoietic stem cell is mostly will grow to the bone marrow and spleen, and this liver will have the more support for the metabolism of the adult mice. So I'm studying the effect of the specific genes in the hematopoietic stem cell progression in the embryonic stage. So one of my interests is the NF-kappa B signaling, which is the very important cytokine receptors signaling pathway, which is involved in the many cases of the normal as well as in the tumor and inflammation. So this actually this signaling pathway will have the two arms, which is the first arm, it will induce the activation of the NF-kappa B signaling and MAP kinase signaling, which is it can induce the more survival of the cells, which is the other arm actually is this TNF receptor. When it's bound to the TNF in the case of inflammation or other, it will also can cause the apoptosis or necroptosis of the cancer cells. So actually it has the two arms. It can induce the proliferation, but it also can inhibit the proliferation of the cells or cancer cells. So there is many component of this signaling pathway actually. But interestingly, like the during the embryonic development, actually when we delete this specific gene from the signaling, actually it will cause the embryonic lethality of the mice. So when they are usually when they are in the intact form, they will grow to the adulthood. However, when we knock out this gene or we delete this gene, they will die in the embryonic level. For example, in the embryonic days 14, 16, and etc. And some mice will die after they birth, soon after they birth, and then even some of them also they only survive for several days after birth, but unfortunately they will die. So we are really curious what is actually affecting this cell death, like what actually happened when we knock out the cells and this gene. So yeah, as I mentioned previously, the activation of the NF-kappa B in this gene is my focus is in the P65 and IKK2. This is P65, and in mouse it's called as a real agent. These two genes actually very important transcription factor, which is important for the survival of the gene, which is IKK is the upstream of the P65, and P65 is the main transcription factor, which is activates many kind of the cytokines. And once activated or once the TNF binding to this receptor, it will induce the activation of the TNF alpha, and it will create the positive feedback where it can cause the embryonic survival. However, what happens if we knock out this gene or if we delete this gene, actually it will unfortunately it will cause the paradoxical expression of the high TNF alpha, which later actually it's in turn to the overload of the cytokine and more binding to the receptors of the TNF receptors. And then finally, actually, instead of the involving in the embryonic survival, it will cause to the embryonic stem cells inhibition, or it will go to the cell death. So in this case, we are really curious, like, what is the cell responsible for this molecular mechanism? And then what is the specific molecular cues behind this TNF alpha amplification, which is result in the embryonic survival inhibition and then the stem cells inhibition. So actually, this is the new advance in the stem cell and cancer study. So using the single cell RNA sequencing and then the spatially resolved transcriptomic analysis. So the single cells means like for example, in here, because I'm studying about the liver development in the fetal of the embryo. So I will take the liver and then I will create the dissociation from the fetal liver so that they can have like the suspension of the cells, the mixture of the cells. And then using this technology, we will be able to see specific clusters of the cell, which is this actually one dot means for one specific cell types, and these clusters color will tell us that this is the same cell cluster. So for example, in here, like we have like more than 5 clusters from the liver cells. And the other thing is also the spatial transcriptomic analysis. Actually, it's similar with the single cells. However, in here, like in the single cell, actually, we dissociate the sample, so the cell will become the suspension. However, in the spatially, we will take the whole embryo or maybe in the whole tumor cells. We will plate them in the paraffin or the fresh frozen, and then we will cut them in the slide, and then we will know how this cell actually make the cluster. So basically, it's similar. Like we can see in the single cell level, like how's this, what is the component of this liver from the stem cell, or for example, in the tumor cell, if we put like what is the composition of this tumor bulk. However, with the single cell, we cannot know how they interact each other, like where they are actually located in the specific liver site. But with this the spatial transcriptomic analysis, actually we can spatially resolve where the location of the cells. For example, this one, the green one, will have the close proximity with the blue population, etc. So we use this technology to study the TNF alpha and F kappa B signaling in the immune response, cell death, as well as the stem cell progression in the developing liver. So yeah, so what we do is like we will omit the mice, and then because we know that the knockout of these mice will have the embryonic lethality, so we harvest them in the embryonic stage, and then we will do them single cell RNA sequencing as well as the PCM transcriptomic analysis. So yeah, firstly, of course we will characterize how this deletion will involve in the embryonic lethality. So for example, in this one, this is the wild type mice, which is the intact P65 gene, and then this is the knockout mice where we delete the P65 gene. We can see that this one, for example, in the E15.5, actually it's have the smaller size, and then during the E16.5, they have the hemorrhage, or like the liver is bursting with the many blood cells, something like that. And actually during the time, only the wild type or heterozygote mice will born. However, the knockout, they will be lice, they will be die, and then they will not born from the mom. Yeah, so usually we will characterize. So for example, this one we do the staining from the apoptosis marker. So it confirms that in the liver, many apoptosis events are happening in here. And then we will also involve this technology of the single cell RNA sequencing protocol, which is mainly it's like we put the specific barcode on the cells suspension. So each cell will have the specific barcode, and then when we amplify the cDNA and then we do the sequencing, this specific barcode will give us the clue what is the cell type of the specific cells. Yeah. So when we do this assay, we can also check what is the gene expression of the cells, or what is the cell-to-cell interaction. However, in here, like we still don't know whether they are in close proximity or not. Yeah. So this is just an example, like how the analysis looks like. So we will be able to tell, for example, this one is in the liver. We can see like there is a population of the B cell, and then myeloid progenitor on the covert cell, hepatoblast, and etc. And then it will we can get specific like marker of the cancer cells, like cell type population like this. And of course, we will further differentiate between the wild type and then the knockout mice. What is the difference of the population, something like that. And yeah, this is the example of the single cells technology. And then for the spatial, I will give example for the cancer one. So previously in my PhD, I'm studying about the hepatocellular carcinoma. In here also, I still be able to continue my study in the liver cancer or HCC progression. So this cancer actually is very common, and then it's commonly happen in the male and then female, and it was the part of the mainly like main 10 more progressive cancer in the male actually. Yeah, and especially in the Asia, it's more prone to have the liver cancer compared to the Western people. And there is a specific inhibition already. People invented to inhibit this hepatocellular carcinoma progression. However, one of the therapy is not enough. So people are trying to put the multiple targeted therapy by put two inhibitor to the cancer cells and see how they can reduce the cancer progression. But however, there is still no validated the biomarker actually for the HCC, because as you know that the liver size actually is very big. So once you take out the one specific site of the cancer cell, actually you don't know whether it will be the same with the other part of the liver or it will be different. So it's very hard to distinguish what is the predictive biomarker for the HCC. So it's very hard to create the specific targeted therapy also. So what we are doing is like sometimes when the patient we cure already, however, the liver cancer, for example, we will take out the cancer site from the patient. However, often the time, for example, 2 years, like 3 years, they will occur again the tumor or as we call like cancer recurrence. So we will compare like the patient without recurrence, and then with the recurrence, we will take the multiple sites of the liver cancer in here, and we do the single cell analysis, but we will also do the spatial transcriptomic, whereas this one is the site of the cancers. We plot in the transcriptomic analysis, and then from here we can get many information. For example, like what is the cellular density of the tumor niche, and then how the ontogeny and how the colocalization between the biomarker. So we can also know about the immune cell interaction between these tumor cells. And we also do some validation with some of the molecular technology for validation of the specific gene. Yeah. So similarly with the single cells, we also projected this for the sequencing and then mainly by the bioinformatic analysis. So yeah, we are able to collected several patient sample in here, and then we use them as the spatial transcriptomic analysis. And then we have several sections also in here. From this one patient, we have 5 sections, or sometimes it have the 4 only. And then we have the recurrence also, and then non-recurrence, and then we will compare each of them, how like the spatial distribution of the biomarker of the cancers. Yeah, this one is mostly the bioinformatic analysis involved. Yeah, I'm not bioinformatician, so I will not talk about this one. So mainly like using this technology, actually we can know within the tumor cell environment, what are the cells that are interacting together. For example, in here we can see like the red and blue color are close. Then where we annotate them, like, oh, for example, this one is the dendritic cell. For example, this one is the tumor cells. Then the specific tumor cell are very close interaction with the dendritic cell or something like that. And we also can also correlate with the pathologist analysis, like whether this area actually represent the correct tumor site or no. And the benefit of this technology is like we can know what is specific cell type that is more active in the cancer cells. For example, in here, like we found specific endothelial cells, which is actually is also normal cells, but also in the cancer, it will invade to the microenvironment, and actually it will cause more progression of the tumor cells. Yeah, and we do also similarly like in vitro studies. Sometimes we do also the gene analysis, like what is the gene that is involved in the cancer progression, and then we can do the ligand receptor interaction also, how this cancer cell interact with the other ligand or receptor from the immune cell or others. And then usually we will validate our result also by the staining, like how this cancer is, how this specific population actually is overexpressed in the specific cancer, in the recurrence and without recurrence, something like that. And yeah, long story short, like we find by this technology, actually, we can know what is the cell clustering, and then we can know what is the cell-to-cell interaction and ligand receptor interaction, as well as we can know more about the molecular mechanism, as well as how it's really affect the cancer prognosis. So yeah, this is the advantage of the current cancer and then the single stem cell study. However, like this single cell and spatial actually is quite costly. Like, yeah, it's cost like more than $1,000 per sample. So sometimes it's quite challenging, but actually it's also provide very informative area of very informative information for us. Yeah, so far that is something that I can share. So thank you so much for your attention. Yeah, I hope I didn't give too much information. I hope I didn't make you confused. But anytime you have question, you can ask me.
Okay,
Thank you
thank you
so
so
much.
much, Miss Ita, for that amazing and inspiring presentation. It's really exciting to hear about all the research you're involved in, especially when it comes to cancer and it could change the future of cancer treatment. So let's open up the question and answer session. If you have anything you like to ask Miss Ita, feel free to raise your hand on Zoom platform, or you can drop your question in the chat box. Let's take this opportunity to learn even more with Miss Ita. And if you're not confident enough to ask the question in English, you can ask the question in Bahasa instead. It's totally fine. Okay. Okay. Is there any question? Or maybe while we wait for question from the participants, may I a question for myself from myself? Is that okay?
Sure.
Okay, so earlier, Miss Ita, you mentioned about chemokines and the tumor microenvironment, right? So
Yeah.
I'm curious about how chemokine receptors on stem cells affect their interaction with both the bone marrow niche and the tumor microenvironment. And then is there any specific chemokine receptor found only on hematopoietic stem cells, and how do these receptors influence hematopoietic stem cells movement within the bone marrow niche or during cancer spread? Thank you.
Yeah. So actually, the beauty of the cancer cells actually because they have the capability to act abnormal. So that's why it's supposed to act like normal, but it's having the specific behavior. For example, instead of the promoting the correct metabolism, it will create more like cancer progression. So it's also very interesting because sometimes like they have the mutation where they cannot like act how they do, or there is like specific external factor which is also adding the more like cancer carcinogenesis factor, which is they can act separately, something like that. And sorry, what's the second question? The blood
Okay.
cancer.
The second question is, how do these receptors influence hematopoietic stem cells movement within the bone marrow niche or maybe during cancer spread?
Oh yeah. So mostly like if it's affecting the movement, usually it will react to the receptors, which is important for the opening of the blood barrier. So because at first, the cancer movement, it will extravase to the blood vessel, right? So they will influence this membrane integrity. So they will mostly like working with the receptors important for the extracellular matrix. So they can involve to the invasion to the other part of the body. Yeah.
Okay, thank you so much for the answer, Miss Ita. Okay, I see someone has raised their hand. Okay, maybe the first one is from Mr. Andigana Kusuma. Please go ahead and ask your question.
You are mute. Sorry.
Okay. Mr. Aan, you're still in mute. Maybe you can unmute yourself first.
Okay.
Okay.
Okay. Okay. Okay. I want to say hello with my old friend Ita.
Hi, Aan.
It's like a reunion. Yeah. Maybe you are the greatest symbian. Yes. Symbian stands for Symbiosis Mahasiswa Biologi 2008. Welcome back, Ita. We are very proud of you. Okay, I have some question for you. The first, based on your point of view, how about the research cancer in Indonesia? It will be compared with in Korea, in Singapore, and in the other country. And the second, if we want to, especially as a biology student, if we want to study furthermore about cancer, what the basic skill that we have to do as a student biology. And then the third question is
Okay, can I answer first before I forget?
okay, okay. Yes,
so,
please. Okay, okay.
Yeah, for the first question, for the cancer study in Indonesia, basically I can say that actually I'm not sure how people think, but I mean like in Indonesia actually the cancer research is not really supported by the government. As you can see, actually, as far as my understanding, like for example in Korea or in Singapore, the government are really supportive to give the research fund for the cancer research because they know like this cancer is a serious disease that need to be eradicated. So they will invest a lot of money and then they will invest a lot of education for this section. However, I feel like in Indonesia, sometimes like it's like they study the cancer also like some institute like biopharma or some institute like I3L. I know also they study about the cancer. However, like they have the limitation because first of all, like the cancer study sometimes require big cost. So like the school or the research institute cannot afford with the like the technology or something like that. So yeah, I think like in Indonesia, like we need more support for the cancer study. Like I will be happy, like I mean to work in Indonesia also. However, if there is no support, it's also like actually is not a good sign also. So yeah, but yeah, it's especially like Indonesia is also research. I mean like the cancer event actually is quite high also. So I think it's deserved to be improved in the future. And then the second, sorry, I have short memory.
The second question, yes.
What is that? Sorry.
The second question is what the basic skill that
Oh
we
yeah,
have
yeah.
to do.
So mostly like we have to have the skill from the cell culture or molecular biology, for example, like for the gene editing, for example, like to make the mutation or something like actually it's just a simple PCR or like plasmid isolation. Yeah, we need to have that kind of the skill, I think. So, but I'm not sure. Maybe in Undip we have the cell culture uh, technology or no. So yeah, it will be good if we have that technology. So yeah, basic like molecular biology such as like the cloning is definitely important. Yeah, Western blot, qPCR, yeah, that's okay. But like yeah, genetic engineering and as well as the cell culture is a must, I think. Yeah.
How about with animal handling, maybe?
Yeah, animal handling like uh mostly like because in my case also I study animal handling in my master course, so it's okay to not have it, but it's better to have as soon as like if you can handle it, it's will be better.
Okay. And the last one. Okay. Based on your research, which one the type of cancer that to be difficult to understanding, or it's all of the cancer is clearly to understanding?
For me, I think the brain cancer is a bit hard. I didn't study because it's very like tricky, because like for example like liver or for the blood cancer is like you just take from the bloodstream or something like that, or the liver is located in the like middle of your abdomen is okay, but the brain cancer is because of the structure of the brain, like it have the thorax also, and then it have the blood brain barrier also, which is it's very actually important. It's very difficult to be like penetrated by the anti-cancer drug. So I think the most tricky one is the brain cancer. Yeah.
Okay, thank you very much. Maybe we can meet in the other chance.
Thank you.
Okay, thank you for your question, Mr. Aan, and thank you, Ms. Ita, for the response. So let's move on to the next questions. Here's one from Kalista Ginting. Please go ahead and ask your question.
Okay, thank you for the chance. So, the disclaimer first, I'm sorry. I apologize for unable to turn on my camera due to some circumstances. But yeah, thank you for Miss Ita for the knowledge that you gave to us. That was so interesting and also so inspiring. So my name is Kalista Clara, and I'm one of the students from biology department. And I want to ask you this question. It's a little bit basic actually, but still, I still want to ask you this. So you say, so as you mentioned before, for methods used in cancer and stem cell studies, researchers, which is you, often perform some cancer growth assays, analyzing proliferation and colony formation in vitro or outside the body. So my question is, like, how is this in vitro study conducted? Like, how are the cells obtained and analyzed? Like, are cell cultures involved in this process? If it's yes, then how is the procedure to do that? And also, I heard that you mentioned about biomarkers used for identify the CSC or cancer stem cells. Like, how do you obtain all of the biomarkers of that? Like, how is the procedure to do that? Okay, that's all. That's all is my question. Thank you so much.
Yeah, thank you for the question. So for the first question, like how we do the in vitro. So in vitro is basically like, for example, usually like we will get the cells or cell line, which is for example, breast cancer cell line or colon cancer cell lines or brain cancer cell line, which is it's derived from the specific type of the cancer. So usually, like there is a company who is like also like working with the hospital. They will get the tumor from the patient, and then they will grow it in the like 3D plate. You know, like if you culture the bacteria, you will grow in the petri dish, something like that, right? So this cancer cell or like in vitro culture is also similar. Like we grow the cells in the dish like that, and we put the media specific for their culture. So for example, like from the brain, we will put them with the supplement, which is important for their growth also. However, like for example, in the bacteria culture, you will see like the colony growth of the bacteria. But in the cell culture, you will see like the cell is getting flattened, like it will have the, for example, like the star flattened form of the cells to the plate, something like that. So this specific cell type usually will be different from each type from the type. And the colony formation also same. Usually we get from the cancer cell bank, the cell line, and then we will grow it into the matrix gel or like kind of the gel for the cancer progression. But differently with the culture dish, this colony forming they will have the clump like colony actually, like we'll make the big clump to the gel, something like that. And then the second question about the biomarkers. So how we obtain these specific biomarkers. So usually what people do is like they will collect the cancer cells from the patient, for example, and then they will do the RNA sequencing to know what is the most specific upregulated gene of the cancer. And then, for example, in the breast cancer, they will found that this specific BRCA1 gene is highly expressed to the breast cancer. So that's why where the gene is highly expressed, they will consider it as the biomarker. That's how the system usually go. Yeah.
Okay, thank you so much for the answer,
Thank
Miss
you.
Ita.
Okay, thank you for the question, Kalista, and thank you for the answer, Miss Ita. Okay, there's another participant that has raised their hand. Diki, please go ahead and ask your question.
Sebelumnya saya berterima kasih sudah diizinkan bertanya. Saya akan bertanya dalam bahasa Indonesia ya, Miss Ita. Miss Ita, jadi saya pernah melakukan penelitian tentang KP saya itu di Bio Farma di bawah naungan Bu Ayu. Itu mengenai studi in vitro pada model acute lung injury atau ALI menggunakan sel A549 atau epithelial lung cancer carcinoma. Nah, jadi di situ tuh saya melakukan terapi menggunakan sekretom dari mesenchymal stem cell yang diinduksi oleh beberapa konsentrasi HGF. Itu menunjukkan bahwa pada dosis 8 nano HGF itu menunjukkan terjadinya penutupan luka pada A549. Jadi saya menggunakan metode scratch.
Sorry, suaranya hilang.
Diki, mungkin bisa diulangi? Suaranya tadi hilang.
Halo, halo.
Suaranya masih hilang. Oke, ini sudah ada.
Oke. Yang ingin saya
Diki, sorry, suaranya hilang lagi. Masih hilang. Mungkin bisa di type di chat box. Oke. Maybe mungkin sambil menunggu Diki, let's move on to a question from the chat box first, I think. There's one from Puspahning. I will read it for you. Dear Miss Ita, thank you for the interesting lectures. I'm interested in your research about sgRNA sequencing and transcriptomics. My question are, what is the special relationship between GSGS and immune cells, and how does this interaction affect tumor immune evasion? And the second question is, can sgRNA sequencing help predict resistance mechanism?
Okay, so for the first question, what is the spatial relationship of cancer cells and immune cells? Oh, so, okay. So the cancer stem cells are usually having the resistance for the immune cell recognition actually. So usually what will happen is that from what of my observation from the spatial transcriptomic, for example, in the liver cancers, so this specific cancer stem cell, they will have specific like ligand inhibition to the immune cell. So actually, when the immune cell recognize the cancer stem cell and the tumor cells, they will kill these tumor cells. But however, when the cancer stem cells actually exist, it will have the capability to modify, like make the mutation of this ligand receptor interaction, by which the immune cell will not be able to recognize this cancer stem cell. So mostly using the spatial transcriptomic, we will know how like this immune and then cancer stem cells, like the ligand receptor interaction is happen, because mostly like the immune cell, like they are using their ligand or the receptor to act for the killing activity. So yeah, mainly we will use this one to study what is the relationship by the ligand receptor analysis. Yeah. And then the second, can single cell RNA predict resistant mechanism? Yeah, actually it's possible because we're using the single cell RNA sequencing, we can know like which, for example, which specific of the cell types is very high expressed. For example, like macrophage cells, it can also induce the cancer autoimmunity, which is they cannot be targeted by the anti-cancer drug. So the existence of the specific cell type, it can predict the resistance of the cancer itself. And from Nikki.
Oke, from Diki, apakah terapi stem cell atau terapi sekretom dapat membantu perkembangan sel kanker itu sendiri, dan apakah stem cell dapat berubah menjadi sel kanker?
Okay, so bahasa ini, okay. Coba
Okay,
bahasa
you want to answer in Indonesia, Miss Ita?
Ya, jadi sebenarnya kayak si stem cell ini kan fungsinya untuk regenerasi ya. Jadi kadang bisa juga kita terapkan terapi stem cell untuk the cancer cells, tapi harus benar-benar yang di stem cell tuh harus apa namanya? Sorry, I think I'm more. I just talk in English. I think because this stem cells sometimes yeah, as you can see, like it can grow to the cell, cancer stem cell, but it's actually something dangerous. So what happened is like previously also in South Korea, actually, like one of the PI actually tried to develop the anti-cancer treatment using the stem cell. However, like when they use the cells, actually it's changed to the cancers. So basically, I think that this secretome from the stem cell, actually, it does help to reduce the cancer progression. For example, if we put the stem cells, which is important for the immune system, then it can help to treat the cancer. But however, if it's changed to the cancer cell, they will not use this in the clinical trial in the future. Yeah. I hope I answered your question.
Thank you, Miss Ita.
Okay, thank you for the question, Diki and Mbak Puspahening. Okay, the next question is from Wigeni Ayuning Tias. Thanks a lot, Mbak Ita, for the amazing lectures. I'm curious, is that any commercial stem cell product in Singapore? Thank you.
Yeah, I think so. But so far, like, it depends like what product you want. Like, for example, if for research, like for the stem cells, cell growing, the company also selling. Or some people do like the stem cell therapy for the skin, something that people also selling also in Singapore. And recently also, they have the stem cell therapy also for the cancer patient. However, like because of this stem cell treatment is actually quite pricey or expensive, so not many people can buy this treatment actually. And even the insurance actually, they will not cover for the stem cell or immunotherapy system. So actually it's exists, yeah, but it's very pricey.
Okay, thank you for the answer. Is there any further question from Wigeni Ayuning Tias, or is it enough? And maybe there's one more question in my direct message. It's from Fahira. Ma'am, I have a question. You have been told that someday there are drugs that can inhibit the gene. So my question is why we have to inhibit it? Why not curing? And is there any side effect if we just always inhibited it for too long? Thank you.
Why not what? Sorry.
Why not cooking it? And is there any side effect if we just always inhibit?
Yeah, that's true. Sometimes if we inhibit, like it will have the side effect, like for example, like it will affect to the others, like osteoporosis or something like that. Like it will have the specific effect. However, like actually, like inhibition, I cannot say like it's 100% cure, because like once we inhibit the cancer cell, actually like this drug is not really enough, or it can also kill another type of cells, which is the side effect also, which is because it's actually, you know that most of the cancer patient that have the chemotherapy or the drug treatment, because they are targeting the proliferating cells, but because of the hair is keep regenerating, they keep growing and proliferating, they will also have the hair loss. So that's also the side effect of the drug. Yeah, I think it's true, like why we always inhibit, why not improving the quality of life? For example, like improving the quality of the cell, specific cell type or something like that. That's why like people usually in this day are combining, like not only using the targeted therapy, using the drug that inhibiting, but usually also sometimes using the natural product, for example, like ginseng extract or something. People also study how it can improve the immunity of the human body, so it can go to inhibit the cancer progression. So yeah, one inhibition actually is still not enough to cure the cancer. So we need multiple inhibitor, because like the cancer cell is very unique. So I'm agree with you. We need to improve both like inhibition, and then also improving the like the immunity of the normal cell itself. I hope I answered your question.
Okay, any further question maybe from Fahira, or is that enough? Okay. Maybe there's another participant that will ask a question for Miss Ita. Okay, or maybe there's no other question again. Okay, there's another question from Wireni Ayuning Tias. Is there any specific biomarker between stem cell and stem cell cancer?
Yeah, usually it's will be different. So the biomarker for cancer stem cell, it will be unique one from the normal stem cells. So this biomarker, when we target, it will only target the cancer stem cell, but not target with the normal stem cells.
Okay, any further question from Miss Wireni Ayuning Tias? Or is that enough? Okay, maybe one more question for the participant. Okay, thank you, Miss Wireni, for the question. Maybe one more question for the from the participant. Okay. I believe there are no more questions for this session. There have been so many questions, right, tonight, and I'm glad to see such an interactive discussion. Thank you so much, Miss Ita, for the insightful presentation and for addressing all the questions today. Your expertise has truly helped and deepen our understanding on stem cell and cancer treatment. On behalf of everyone here, I'd like to express our appreciation for your time and valuable contribution. And maybe before we close this session or this event, I'd like to ask Miss Ita for your thoughts on tonight's discussion, and perhaps if you have any advice or message for students in the biology department who may be interested in pursuing stem cell research.
Yeah, it was a very wonderful night for me having this talk. Actually, I didn't expect it will be a lot of people. Like, yeah, usually I just talk in front of few people, but it's very good to share my experience also. And also for the students in the FSM also, I hope like you guys also keep growing your interests in the research. Like, I mean, it's a good investigation to study in the research actually. So I keep encouraging you to keep study, and then if you like want to pursue to a higher degree, it will be better. like it will improve your knowledge, but also it will help to help our research in Indonesia in the future also. I believe in that. So yeah, and if you have any question or something, or like about the study or about the daily life also, you can ask to me also. So actually, previously also one of the junior in Undip, which is, I don't know, is 2000 something. Actually, she also asked the information from me for the scholarship also in South Korea, and then she also follow me to have the study in the South Korea also now. And now she's graduating already. Her name is Yu Saratu. So she also get the master degree from South Korea, and then she also now I think working in Jakarta already. Yeah. So I know that maybe in Indonesia, like not many of the cancer study is open, but I believe like if we have more interest and then there is support from the university and then from the government, I believe that we can make this studies more wonderful, and then we can create something more useful for the not only for ourselves but also for our community, for the cancer patient, as well as for the Indonesian education and then health also.
Okay, thank
Thank you
you,
so
Miss
much for
Ita.
having me.
Okay, thank you for Miss Ita for your time and your contribution. I hope we can meet again in the future. It will be nice to connect further, or maybe I will reach you out later regarding my PhD program. Is it okay?
Sure, sure, sure.
Okay, okay. I also want to thank all the participants as well for your engagement. Now I will hand it back to the Master of Ceremony to close the session. Thank you once again.
Thank you.
Okay. Yeah, thank you, Miss Rizky, for moderating the session so excellently, and thank you for Miss Ita for the wonderful and insightful presentation, lecture, and also the discussion as well. I would also like to thank all the students for your participation in this lecture session and also in the Q&A or discussion session. And before end this event, please don't forget to fill out the attendance form, which has been shared in the chat. So last but not least, let's give a virtual round of applause to our speaker and moderator. Maybe you can react through the Zoom. On behalf of the entire committee, please forgive us for any mistakes during hosting this today's general lecture. Thank you for attending this event. See you. Have a good rest, good night, and wassalamualaikum warahmatullahi wabarakatuh. Thank you again, Miss Ita, for
Yeah, thank
joining
you
us
also.
in this event.
Yeah. Good night.
Okay,
Good
good night,
night.
Ita.